Today, I'm going to talk about the signal molecule dopamine, its transduction pathway, and the cellular response it evokes. Dopamine follows two main types of cell signaling, synaptic signaling and endocrine signaling. Today, I'm going to cover synaptic signaling, which is how dopamine communicates between neurons. Dopamine is a neurotransmitter released by neurons in the brain that originate in the midbrain, primarily the ventral tegmental area and the substantia nigra. When an electrical signal or action potential reaches the axon terminal, the neuron prepares to release dopamine. Let's take a closer look at the synapse. When the signal arrives, dopamine-filled vesicles fuse with the membrane and get released into the synaptic cleft. When activated, the D1 receptor turns on a nearby G protein. The G protein splits, and the alpha subunit carries the signal deeper inside the cell. The alpha subunit activates an enzyme called adenyl cyclase, which produces the second messenger, cAMP. D1 receptors stimulate the adenyl cyclase, so this receptor basically controls cAMP levels. Protein kinase A, or PKA, is a crucial enzyme that translates dopamine signals. High cAMP levels from the D1 receptor activate PKA strongly. PKA then phosphorylates ion channels by adding a phosphate group. This phosphorylation makes the ion channels open and positively charged ions rush into the cell. This is called depolarization. This makes the inside of the neuron more positive, which brings it closer to sending its own action potential. When enough charge enters, the neuron fires a signal, and the result is an increase in activity in the brain involved with movement, motivation, and reward.